তুমি লাষ্ট ক্লাছৰখিনি লৈ লৈছা আজি লৈছা Okay, last class, we discussed, we were talking about the classification system, right? 2 kingdom, 3 kingdom, so we were talking about the kingdom Monera, or Monera basic characteristics. So we will be talking about one type of Monera, that is eubacteria in details, heading koriba eubacteria, likhiba structure of bacteria, diagram. যোৱাকালি কি পঢ়াইছিলোঁ যে when we talk about the kingdom Monera, Monera কিমান types of Monera আছে 7 types of Monera আছে তাৰ ভিতৰত one type is eubacteria. Eubacteria is also called as true bacteria, right? So আজি কি কৰিম So what we are drawing is the structure of a bacteria. Whenever we talk about bacteria, if you all have studied, we know shapes of bacteria. So bacteria dekha dekhi nohoy, right? We have 4 basic shape. One, sometimes the bacteria looks spherical, sometimes the bacteria look like a rod, sometimes it look like a comma, or sometimes it looks spiral, right? So different forms of bacteria ase. Ami kuntu shape akiso, ami rod shape bacteria akiso, right? It looks like a rod, right? Why are we drawing a rod shaped bacteria? Because the most common type of bacteria that we see is a rod shaped bacteria, right? So structure of bacteria jetia akiso, bacteria is nothing but an example of prokaryote. So this could be the structure of a prokaryotic cell also. We are going to study a chapter called as cell, the unit of life, where we have to talk about the structure of both eukaryotic and prokaryotic cell. But since we will discuss the prokaryotic cell here, we will not be discussing at that time, right? Cell unit of life or okol ami eukaryotic cell to kore, karon prokaryotic cell or structure already ami akilwe, right? Look into the features and draw the diagram. So once it's over, we'll explain. এইটো লিখিব পাৰা, it is together called as cell envelope. Nuclear, circular DNA, histone protein absent. Ribosome. 70S ribosome. over? Okay. So what we are drawing here is we are drawing a structure of a prokaryotic cell, and the example that we are taking about a prokaryotic cell is bacteria. Whenever we talk about a bacteria has a cell envelope. Cell envelope is the layers, right? Jod outermost layer would be called as capsule, right? The next layer would be called as cell wall, then the innermost layer would be called as the cell membrane. Have you heard the word called as plasma membrane? Plasma membrane শুনিছা যিকোনো এটা সেল আইদার ইট ইজ আ সেল প্লান্ট সেল অর এনিমেল সেল এখন বাউন্ডারি থাকিব ন বাউন্ডারিখনক আমি কি বুলি কওঁ ইফ ইট ইজ আ প্লান্ট সেল বাউন্ডারি কেইখন থাকিব দুখন থাকিব সেল ওয়াল আৰু সেল মেমব্ৰেইন থাকিব সেল মেমব্ৰেইন ইজ অলসো কল্ড এজ প্লাজমা মেমব্ৰেইন এণ্ড ইফ ইউ আৰ টকিং এবাউট এ এনিমেল সেল তাৰ সেল ওয়াল নাথাকে কিন্তু প্লাজমা মেমব্ৰেইন থাকিব ৰাইট সো বাই দ্য ওয়ার্ড সেল মেমব্ৰেইন we understand we are talking about the plasma membrane so এটো হল capsule inside the capsule this portion would be called as কি cytoplasm right in the cytoplasm what we will have is a different organelles first we will have the genetic material so eta prokaryotic cell or eta eukaryotic cell could be divided into two types prokaryotic and eukaryotic one basic difference between the two ki hoy one difference between a prokaryotic cell and a eukaryotic cell agote khunisa prokaryote or eukaryote right so difference ko eta prokaryotic cell eta example bacteria eukaryotic cell example animal cell amar cell hobo pare right so what cell wall is the basic difference between a plant cell and an animal cell right kintu prokaryote or eukaryote er majo basic difference kwa presence and absence of the nucleus right eukaryotic cell or nucleus thake prokaryotic cell or nucleus na thake jedi nucleus na thake does that mean tar genetic material na thakibo obviously genetic material thakiboi libo so in case of a prokaryotic cell we will not see a nucleus instead we will be observing a structure that is called as nucleoid nucleus or nucleoid or difference ki jedi genetic material khini eta membrane or bitor mo ase na if you talk about a nucleus or ki thake ekhon membrane thake dukhon membrane thake tar bitor genetic material khini thake right jedi membrane thake ta ami ki boli kau nucleus boli kau kintu prokaryotic cell etat organelles bilak ki hoy membrane less organelle hoy to obviously nucleus or to ki na thake membrane na thake so such type of nucleus would be called as ki nucleoid so first point is genetic material in the form of nuclear by bacterial genetic material or naam ki nucleoid it has ki nucleus or bitorot genetic material thake genetic material in the form of ki genetic material in the form of DNA RNA etc right so in this case when we are comparing nucleoid and nucleus the first point that we have said that nucleoid ko thake prokaryotes eukaryotes ki thake nucleus thake the second point nucleoid ki na thake nuclear membrane na thake nucleus or nuclear membrane thake nucleus eta bitorot genetic material thake nucleoid to genetic material ke ta mane in both the cases DNA is present if you are talking about DNA no structure of the DNA in case of a prokaryotic cell the structure of the DNA is circular in case of a eukaryotic cell the structure of a DNA would be something like this is a point clear it is structure ami class 12 or pohim goi so bacteria type key type of DNA thake circular DNA is a word that you have to remember clear okay next is together with that tahator ki thakibo ribosomes o thakibo when we talk about ribosome ki hoy eta cell organelle hoy and it is responsible for ki it is responsible for the formation of proteins right jehetu eta living cell tahator to proteins lagibo so proteins kune banabo ribosomes whenever we talk about ribosomes basically there are two type of ribosomes one is 70S or one is 80S 70S is a characteristic of prokaryotes prokaryotic cell etat kenukwa dhoroner ribosomes thake jikono prokaryotic cell etat 70S type of ribosomes thake eukaryotic cell etat both 70S or 80S thakibo pare right okay so they will be having 70S ribosomes we understood the cell wall next they will have certain granular substances like volatile granule glycogen granule etc grain like substance so eblak granules of ki thakibo mane they will be storing phosphate food etc next is this is understood we'll come to this one we have something called as a gas vacuole vacules ini pao no whenever we talk about a plant cell, no, one difference between a plant cell and animal cell, plant cell or ki thake eta centrally large vacuole thake, animal cell or na thake, right? Bacteria ki thake vacuole present ne absent, actually sabole gole vacuoles are absent in bacteria, right? But we have gas vacuoles okol kot thake aquatic forms, karon bacteria গোটেই ব্যাকটেৰিয়াবোৰ একুৱেটিক নহয়তো, কিছুমান লেণ্ডত থাকে ব্যাকটেৰিয়াটো আমি কালি লিখিছোঁ ইউবিকিউটাছ হয় যদি পায়, ৰাইট? চ' যোনবিলাক একুৱেটিক বেক্টেৰিয়া থাকে তাহাঁতৰহে কি থাকিব, গেছ ভেকিউল থাকিব, এণ্ড এজ এ নেম ছাজেষ্ট ভেকিউলৰ ভিতৰত কি থাকিব, এয়াৰ থাকিব, আৰু এয়াৰ থকাৰ কাৰণে ইট উইল হেল্প ইন ফ্লোটিং, ৰাইট? এণ্ড দ্যাট ইজ হোৱাট ইজ কল্ড এজ বুয়েন্সি। নেক্সট ইজ these vacuoles are also called as ki pseudo vacuoles, tamane vacuoles absent kintu pseudo vacuoles present, clear? Okay, next is ekta structure pisot jam. Apart from the genetic material, bacteria cell etar genetic material ki hoy, nucleoid, tat ki thake circular DNA. Apart from this genetic material, they will be having a extra genetic material that is called as plasmid. What is plasmid? Extra genetic material in case of a bacterial cell. Talking about plasmid, ami plasmid our definition likhim olop pisot. Plasmid could be different types in different bacteria. Right? Next is we have hair-like structures in the diagram. Whenever we talk about hair-like structures, kimanta hair-like structures, there are 3 types of hair-like structures. We see something called as pili, fimbri, or flagella. The first point, whenever we talk about hair-like structure, all hair-like structures are not responsible for locomotion. Generally, আমি যেতিয়া hair-like structure দেখা পাও, no, for example, cilia hok, flagella hok, এবিলাকৰ কাম কি? Locomotion. কিন্তু in case of a bacterial cell or prokaryotic cell, flagella is only responsible for locomotion. Pili and fibri are not involved in locomotion, right? Function, both of them, attachment and conjugation বুলি কিবা এটা বস্তু আছে, মই পঢ়াম অলপ পিছত, right? So we have discussed almost all the parts except something called as a mesosome, right? So in a bacterial cell or in a prokaryotic cell, we see another structure that is called as mesosome. Mesosome is the place or is the area. What is mesosome? Eta word jodi use koro etu buzi paba ne soason. এই ওয়ার্ডটো বুঝি পাও নেকি? ইনভেজিনেশন। ইনভেজিনেশন মানে কি? সোমাই অহা পোর্শনটো, right? So what is a mesosome? Invagination of the plasma membrane. ভিতৰৰ সবতে ভিতৰৰ মেমব্ৰেনখনক কি বুলি কৈছে? সেল মেমব্ৰেন বা প্লাজমা মেমব্ৰেন। প্লাজমা মেমব্ৰেনৰ এটা পোর্শন কি হৈছে ভিতৰলৈ সোমাই আহিছে, that is what is called as the mesosome. First point. Second, এই সোমাই অহাৰ কাৰণে প্লাজমা মেমব্ৰেনখনৰ সাৰফেস এৰিয়া বাঢ়ি গৈছে নাই? যদি স্ট্রেট হৈ থাকে অকল এইখিনি সারফেস এরিয়া তাহাঁতে অকুপাই কৰিছে, যদি সোমাই আহে তেতিয়াহ'লে তাহাঁতে লাৰ্জাৰ সারফেস এরিয়া অকুপাই কৰিছে। সেকেন্ড পইণ্ট। থাৰ্ড পইণ্ট ইজ হোৱাট ইজ দ্য ফাংচন অফ আ মেসোসোম? It helps in respiration. Karon whenever we talk about, for likham olop pisa, whenever we talk about a eukaryotic cell, no, eukaryotic cell eta respiration coat hoy boli kole ki koba? Eukaryotic cell. Eukaryotic cell etat respiration hoy no, obviously hoy living cells jehetu tat respiration hobo. Respiration hole ki form kore? First, what is respiration? Or the question could be, we know cells respire. Why do cells respire? Amar to protect the cell ote ki hoy respiration hoy no, cellular respiration hoy. Cells bilake respire kyo karne kore? Energy karne. Energy in the form of ki? ATP. Have you heard about this word called as adenosine triphosphate, right? So jete respiration hoy tahatok ki lage? Energy lage. Energy ki form kore? ATP form. So why do you respire? To get energy in the form of ATP, right? Next point, respiration coat hoy by ATP formation coat hoy. Heard about something called as the powerhouse of a cell? Mitochondria, right? So mitochondria powerhouse of a cell boli kiya koise? ATP produce hoy. Respiration kiya hoy? ATP produce koribole. ATP code produce hoy mitochondria hoy. So protekto eukaryotic cell or mitochondria ase, respiration o hoy ar ATP form hoy, right? Hetu hol eukaryotic cell. Prokaryotic cell or respiration hoy noy, obviously hobo living cell jihetu respiration hoy tahator ATP form koribo nokore, koribo kintu code form koribo. Prokaryotic cell or mitochondria ase niki? Nai. আছে জানো? Prokaryotic cell or membrane bound cell organelles নাথাকে, মেমব্ৰেইন বাউণ্ড তাৰমানে মেমব্ৰেইন থাকা সেল অৰ্গেনেল নাথাকে মাইটোকন্ড্ৰিয়া বুলি ক'লে হাউ ডাজ দ্য মাইটোকন্ড্ৰিয়া লুক লাইক সামথিং লাইক দিস মাইটোকন্ড্রিয়া ডায়াগ্রাম দেখিছা something like this right এখন membrane থাকিলে না outer membrane inner membrane so membrane থাকা organelles prokaryotic cellot না থাকে তেতিয়া হলে মাইটোকন্ড্রিয়া থাকিব নেকি না থাকে যদি মাইটোকন্ড্রিয়া না থাকে prokaryotic cellot ATP form কৰে so ATP code form কৰিব বা respiration code হ'ব it occurs in mesosomes is the point clear so what is the function of mesosome respiration So, whenever we talk about mesosome or duta function, main function ki respiration, ATP formation. Second function is we have something called as a septal mesosome. Why is it called as such? Because it is involved in septa formation. Septa mane ki wall. Eta cell jete divide koribo duta cell le major ekhon wall lagibo na lage. So that septa kune form kore septal mesosome. Juntu mesosome nuclear genetic material usor thake. হেতুক আমি কি বুলি কম septal mesosome responsible for কি septa formation during cell division এটা cell যদি ভাঙি পেলাই দুটা ফর্ম করে এখন wall লাগিব right okay next this mesosome is called as lateral mesosome lateral মানে কি সাইডৰ পিনে থাকা mesosome টুক আমি কি বুলি কম lateral এটা ইমান precisely question আহা নাই যে what is the function of septal mesosome and what is the function of lateral mesosome question আহিছে what is the function of mesosome right mesosome most important function respiration it also assists in cell division or septa formation is the point clear right so this is the structure etia ami ki korim ei structures bilakor eta point likhim eta ba duta point likhim diagram over heading likhiba plasmid we'll start from plasmid anything to understood na So plasmid और information लिखा Plasmid ok ami ki boli koise extra genetic material why are we using the word extra karon bacterial genetic material to nucleoid asile tar uporiu extra genetic material likhi jaote saba whether you are understanding or not it is not a part of circular DNA is this line understood okay This is something that we did not study, we'll read. So we are talking about plasmid. What is plasmid? Extra genetic material. So this line is important, right? The term was given by Lederberg. It is not a part of circular DNA, understood. Ekhini pitorot, what is important is just F plasmid. Baki kita upor iman questions aha nai. Just to know that there are different types of plasmid. Plasmid ki hoy genetic material. Genetic material jetia tat ki thakibo DNA thakibo, gene thakibo. Depending on kenukwa dhoroner gene ase, plasmid bilakor nambur beleg beleg. Jedi fertility gene thake tak F plasmid buli koy. Jedi resistant gene thake tetal tak R plasmid buli koy, etc. Right? That part is not important. First part is important. Yeah, conjugation porhua nahi no italage porham. Bracket ki likhiso bacterial reproduction. That's a form of bacterial reproduction. Whenever we talk about reproduction of bacteria, the bacteria is a common way of reproduction ki hoy asexual reproduction, that is binary fission, right? Kile apart from asexual reproduction, tahate sexual reproduction dekhai ne dekhai, kintu sexual recombination hoy. One type of sexual recombination is conjugation. We were talking about that process, right? Ekhin loke hol likha hol sobore. Okay, coming to this part, plasmid can be incorporated in the circular DNA or it can be transferred by conjugation. Means what? We are talking about two bacteria. Bacteria pisot ami pohim, right? Etu bacteria tar nijor genetic material ase, etu tar nijor circular genetic material ase. Yate eta plasmid ase, yate nai, right? So a plasmid to le jabo pare, right? So etu jetia yale jabo pare, keneke jabo? A plasmid to it can replicate, right? Duta hobo eta tale jabo goi, or a plasmid to adha part to yale gusi jabo pare. For example, yare adha part gusi gol, right? So ketiaba ki hoy etur pray etule je a plasmid to goal, a plasmid or a part to logot log lagi jabo pare. That is what is called as incorporated, right? If it is incorporated, keneke processor through jabo by a process called as conjugation. What is conjugation? We'll talk about. Is the point clear? Plasmid ki hoy extra genetic material eta bacteria to pa anti bacterial transfer hobo pare. Keneke transfer hobo? By the process of conjugation. So when we talk about plasmid being transferred from one bacteria to another, he ki hobo pare? It has the ability to replicate itself. Replicate mane ki etar pra duta form kora to replicate koy, right? So etu plasmid yale jabo lagile tar duta way ase. Either he duta form koribo, eta yat thaki jabo, eta tale jabo, or eture half part tale jabo, right? Next, jedi half part jai aru a half part to etu bacteria to nar nijor genetic material already ase, ekhini ahi pelai jedi yat lagi jai, then that structure would be called as ki episome. Is the word episome understood? So episome ki hoy jedi half ba eta part of plasmid ahi pelai ba extra genetic material kini jedi circular DNA logot log lagi jai, he structure to kami ki buli kom, he structure to episome. Is the point clear? Now read the sentence and see whether you have understood or not. Clear? It can be incorporated, it can be transferred by conjugation. What is episome, etc. It can replicate, etc. I think it's understood, right? Okay. Next, types of plasmid, F plasmid. Bacteria bilakor, as I said, no, plasmid ki hoy extra genetic material. Depending on ki gene ase, that plasmid to beleg nam. Right? We have something called as F plasmid. F plasmid is responsible for the fertility of the bacteria, and on the basis of this, either the bacteria would be called as a male bacteria or it would be called as a female bacteria. Generally, lower organisms are, I mean, male or female word to use korbo noaru, right? Male something, female something. Hey, male or female word to use korbo lagile morphological tahator male or female structures thakibo lagibo. Human being to male kobo lagile ba human being of female kobo lagile human morphological part thakibo lagibo, female morphological part thakibo lagibo. Jedi nai tel tumi male female kobo nwara. Male female duta ekei bostuk ami kuntuk male kuntuk female consider korim. Eta bostu jete kiba eta bostu ya donate korisil, june donate kore tak ami ki buli kom male, or june receive kore tak ami ki buli kom female. In case of bacteria also, you have two types of bacteria. One is called as male bacteria and the other is called as a female bacteria. Juntu bacteria F plasmid ase tak ami male bacteria ko, and so F plasmid is responsible for fertility. Is the point clear? So F plasmid thaka bacteria bilakok male bacteria, junbilak bacteria F plasmid nai tahatok ami ki buli kom female bacteria, right? Aru male bacteria ta female le ketia convert hobo? Sorry, male bacteria eto suppose male bacteria, eto female bacteria. Yate ki rakhibo plasmid thakibo yat na thake. A plasmid to eta ki capacity ase je beleg eta bacteria ahi jabo pare. Jedi ahi jay eto ki hoy jabo? Male hoy jabo, right? Okay. So this is all about plasmid. Next likha fimbri. So fimbri is a hair-like structure that helps in attachment. So in the diagram that we have drawn, we have seen hair-like structures, some long, some short. The longer one was called as pili, the shorter one was called as fimbri, right? Fimbri or pili kam ki attachment, attachment to solid surface, attachment to mucus layer. Attachment to solid surface is fimbri, mucus layer is pili. Pili is also called as pili qua, pilai qua, g qua, or pilus. It's also called as sex pili or sex pilai. Right? Why is it called as sex pili? Because it is involved in conjugation. Conjugation moi ki kolo one method of bacterial reproduction, right? Olof akote je koisilu je eta bacteria pora antu bacteria le ei plasmid to jabo pare, right? Jabo parile ihote dure dure thakile to jabo nwaribo no. So tahator ki hobo laibo bacteria gita usort ahibo laibo, usort ahi pare tahator edal bridge form koribo laibo. Ei conjugation tube to by a rasta to kune form kore pili bilake form kore hekan tak ami ki buli koiso sex pili buli koiso. Is the point clear, right? So since it helps in the process of conjugation, conjugation is a type of sexual reproduction in bacteria. Who helps in forming this bridge or the conjugation tube? It is the pili, and so the pili is called as sex pili. Next, pili, but this hair-like structure is found in which type of bacteria? Bacteria on the basis of staining. I mean, all of jeti khunisa tomare gram positive, gram negative bacteria khunisa niki? Nai khuna na, I will be studying. So bacteria ki ho on the basis of staining. বেক্টেৰিয়া এটা ষ্টাডি কৰিব বিচাৰিছা বেক্টেৰিয়াটো একদম অকমানি বস্তু এটা কেনেকৈ চাবা চাবত লাগিব ন মাইক্ৰস্কোপ ক'ত থাকে জেনেৰেলি পানীত থাকে চ' পানীত তুমি মাইক্ৰস্কোপত চালে বেক্টেৰিয়েল পাৰ্টছবিলাক ইমান বেছিটো ক্লিয়াৰ নহ'ব চ' ক্লিয়াৰ কৰিব লাগিলে কি কৰিব লাগে ইউ হেভ টু কালাৰ ইট ডাউন ৰাইট চ' বেক্টেৰিয়েল ষ্টেইনিং বুলি এটা প্ৰচেছ পঢ়িম কিছুমান বেক্টেৰিয়াই ষ্টেইনটো লৈ পেলাই ৰিটেইন কৰে তাহাঁতক আমি গ্ৰাম পজিটিভ বুলি কওঁ আৰু কিছুমান বেক্টেৰিয়াই ষ্টেইনটো লয় কিন্তু ধুই দিয়াৰ লগে লগে ষ্টেইনটো নাইকিয়া হৈ যায় দ্যাট ইজ কল্ড এজ গ্ৰাম নেগেটিভ বেক্টেৰিয়া ৰাইট গ্ৰাম নেগেটিভ বেক্টেৰিয়াবিলাকত কি থাকে পিলি থাকে গ্ৰাম পজিটিভ বেক্টেৰিয়াবিলাকত পিলি নাথাকে ইজ আ পইণ্ট ক্লিয়াৰ আৰু গ্ৰাম পজিটিভ গ্ৰাম নেগেটিভৰ কথা আমি আৰু অকণমান ডিটেইলছত পঢ়িম জেনেৰেলি অল দ্য ইনফেকশ্যন ন বেক্টেৰিয়েল ইনফেকশ্যনবিলাক কয় বেক্টেৰিয়া কিছুমানে ডিজিজ কজ কৰে মেক্সিমাম অফ দ্য গ্ৰাম নেগেটিভ বেক্টেৰিয়া আৰ ৰেছপনচিবল ফৰ কজিং ডিজিজেছ ৰাইট সেইবিলাক পিছত পঢ়িম এজ অফ নাও ইজ ইট আণ্ডাৰষ্টুড এনি পইণ্ট ডেট ইউ অল ডিড নট আণ্ডাৰষ্টেণ্ড ক্লিয়াৰ আৰু যোনবিলাক পইণ্ট ইম্পৰ্টেণ্ট সেইকেইটাটো মাৰ্ক কৰিয়ে দিছোঁ helps in attachment in mucus layer ইহঁতেও attachment কৰে ইয়াতো attachment কৰে attachment to solid surface bacteria এটা ইয়াতো লাগিব পাৰে right or mucus মানে কি slimy right পুৰা পনিয়া জেগা নহয় কিন্তু slimy type right ইয়াতে যে solid attachment ইহঁত mucus layer right যেনেকৈ mucus glandৰ কাৰণে আমাৰ কি ওলাই saliva ওলাই ন so that type of area এটা ইনভেজিনেশ্যন ৱৰ্ডটো বুজি পাইছোঁ আৰু এটা পইণ্ট লিখিব পাৰে হেয়াৰ লাইক ষ্ট্ৰাকচাৰ ইফ ইউ ৱান্ট টু ৰাইট কিন্তু ইট ইজ নোন অনলি এইখিনি বুজি পালোঁ চ' ইয়াৰ ভিতৰত ৰেছপিৰেশ্যন ৱৰ্ডটো important, most important word is cellular. When we talk about cell envelope, in a bacterial cell, cell envelope is made up of how many layers? Generally 3 layers. What is the outermost layer? Capsule. Right? So we are talking about the cell envelope. Cell envelope or last layer to ki boli koy capsule, right? Now when we talk about the capsule, not necessary each and every bacteria will have a capsule, right? Kisuman bacteria he outermost layer ba capsule to present hobo pare, kisumanor absent thakibo pare. Jedi absent thake tetia hole cell wall or bahiro pine ki thakibo eta slime layer thakibo. Is the point clear? So we are talking about a bacteria, right? Bacteria etu ki hoy cell wall, etu ki hoy cell membrane, right? So cell wall, cell membrane or bahirat jeti aru eta outer layer thake tak ami ki boli kau capsule, right? So if suppose we are talking about this bacteria where this is the cell wall and this is the cell membrane and the capsule is absent, jedi capsule absent tahator ki rakhiba eta slime layer. Is the point clear? Right? Okay. The next point is, if the capsule is present, তেতিয়াহলে লাভ হ'ব নে লোকসান হ'ব? Obviously লাভ হ'ব, extra protection এটা দিব নিদিয়ে এটা extra layer, right? And a junto bacteria capsule থাকিব, that bacteria would be called as a capsulated bacteria. Capsulated bacteria are always virulent. What is virulent? Disease cause কৰিব পাৰে. কিছুমান bacteria disease cause কৰিব নোৱাৰে, কিছুমানে পাৰে, right? Capsulated bacteria বিলাক কি হয়, যেহেতু এটা extra layer ন, genetic material ক'ত আছে ভিতৰত, তাহাঁতৰ কিমান protection আছে চোৱাচোন, সেইকাৰণে genetic material কিন্তু তুমি easily ভাঙিব নোৱাৰে, সেইকাৰণে সেই bacteriaটোৱে কি কৰিব পাৰে, disease cause কৰিব পাৰে. So capsule provides dash property to the cell. It provides virulent property to the cell. What is virulent? Disease causing capacity is called as virulent. Is the point clear? Sticky character or virulent property ekko noy hi atha lagi dhoribo pare mane jot goise tat lagi dhoribo, you cannot remove it easily, right? But if you are talking about the slime layer, it can be removed easily. Our layers bilak jodi gusai dia, it's easy to kill the bacteria. Is the point clear? We are talking about the layer called as capsule. First point, outer layer. Second point, so bacteria nathakibo pare. Third point, jedi capsule ase tetale hi virulent hobo, hi disease cause koribo, right? Capsule junbilak bacteria nathake tar ki thake slime layer thake, slime layer kon to easily remove kori dibo pare. Over. Desiccation. Oh yeah, kod likhilu, it protects the cell wall against loss of nutrients. We understood. Desiccation is breaking down, right? Drying up. Suppose bacteria bilak generally ki thake pani thake no, pani nuhua jeka bilakot bacteria to mori jabo na, it will dry up. So loge loge jate dry up nokore, loge loge jate mane bhangi na jay, kune protection diye capsule. Next lekha, We are talking about the innermost layer, cell envelope kori asilo, outermost layer ki asile capsule, jedi capsule na thake slime layer. Tinta layer asile cell envelope kon. Innermost layer is called as plasma membrane or cell membrane. The first point, plasma membrane or cell membrane is present both in prokaryotic and eukaryotic cell, and the structure is the same. Eukaryotic cell to plasma membrane ase, prokaryotic cell to plasma membrane ase, structure to eke dekhat, right? As far as the structure is concerned, it is made up of ki, it is made up of phospholipid aru proteins, right? So etu ki akisu, if I am talking about the layer kon jedi tumi magnify kori dia, tele you see a structure like this, right? Jot niki phospholipids bilak thakibo kimanta layer, single layer, double layer. Phospholipid or double layer thakibo aru kisuman dhornor proteins thakibo, right? So plasma membrane is made up of, it is made up of phospholipid and protein. Phospholipid or keta layer, duta layer. He karone phospholipid bilayer. Is the point clear? Whenever we are talking about a single phospholipid, it has two parts. This is the head part, this is the tail part. Phospholipid or etu eta phospholipid, phosphorus ase aru lipid ase, duita log lagi ase. So the structure looks like this. Junto head part hoy, we have used the word called as hydrophilic. Aru junto tail part hoy, we have used the word called as hydrophobic. Eta philic aru phobic mane ki? You like it. Phobic, you don't like it, right? So hydrophilic, like what? Water. So water absorb koribo paribo. Hydrophobic, tahate water to release kori ni diye. Is the point clear? Phospholipid bilayer mane phospholipids or molecules duta layer ot arrange hoy thakibo. Eta phospholipid, etu eta phospholipid, etu eta phospholipid, etu single layer, etu double layer. Phospholipid molecule ase, keita layers of phospholipid ase, duta layers of phospholipid ase, so phospholipid bilayer. Tumi ami sabole gole ami jeta suppose we are talking about a plant cell, right? Plant cell sabole gole ekon cell wall no bahiro kon cell wall. Etu ki hoy? Cell wall. Ekon ki hoy? Cell membrane. A cell membrane kon enlarge korile enuka bostu dekha baba. In case of eukaryote, in case of prokaryote, if I am talking about this as the bacteria, etu ki hobo capsule, ekon ki hobo cell wall aru ekon ki hobo cell membrane. Cell membrane kon dekhat kenuka so eukaryote hok ba prokaryote hok, the membrane has the same structure. Okay, so innermost layer of the cell one understood, also present in a eukaryotic cell, it's understood, right? Mesosomes are present, I think it's understood. If you look into the diagram that we have drawn, mesosome korpra originate korise, cell membrane opera korise, right? Made up of phospholipid bilayer and protein, diagram is understood, right? Coming to this one, semi-permeable membrane, hunisa niki agote? Membranes bilak, membrane jikuno hok, cell membrane hok, cell wall hok, no? Membranes bilak on the basis of ki humabo, ki ulabo, we have a process called as permeability, right? Kisuman membranes of solid, liquid, gas, all the three states of matter in and out koribo pare. Kisumanot okol liquid koribo pare, right? Ar kisumanot GT humai jabo pare, kisumanot choose koribole humabo pare. Based on that process, we have different types of membrane. First, permeable membrane. Permeable membrane konot ki hobo jikunu bostu humabo paribo, jikunu bostu ulabo paribo. Understood? Permeability mane ki? The process of in and out, right? So talking from permeability point of view, ekhon membrane jedi jikunu bostu humabo pare, jikunu bostu ulabo pare, solid, liquid, gas, tetal tak ami ki boli kom permeable membrane. Ekhon membrane jot eku bostu humabo nware, eku bostu ulabo nware, that is called as impermeable membrane. First membrane ki permeable membrane, second ki impermeable membrane, third is we have something called as semi-permeable, right? Etu ki likhisu sosa? Semi-permeable. Ekhon membrane of semi-permeable ketia koba jetia only liquids can enter and come out, right? Yate jeneke gutei states of matter, yate ki hobo only liquid molecules ahibo pare, humabo pare, right? And whenever we talk about liquid, it is not only water that we are talking about. Whenever we talk about liquid, we talk about two words, one is a solvent and the other is a solution, right? Solvent or solution in and out koribo pare. That is called a semi-permeable, tinikhon hol, and the third one is selectively permeable membrane select koribo je ki humabo, right? If suppose there are many substances like X, Y, Z trying to enter the cell, a membrane kon decide koribo muk ki lage. So all biological membranes, almost all the biological membranes are selectively permeable membrane. Hey karone I think you all have noticed je suppose dhora tomarkor mur bikhaise ki kora dorob khwa no, dorob to directly murot na jay, kot jay petote to jay, so petot jo bostu to muror bikh kenege bhal kore কারণ সেই বস্তুখিনি সব সেলছত যাব, সব সেলছত যাব কিন্তু সব সেলছত এন্টার কৰিব নে সিলেক্টিভ সেলটোত এন্টার কৰিব, সিলেক্টিভ সেলটোত, সো দে হ্যাভ আ what semi permeable, sorry they will be having a selectively permeable membrane. I think it's understood. So plasma membrane is an example of semi permeable membrane. So cell envelope discuss কৰি আছো, তাৰ পিছত দুটা layer complete হল, next লিখা cell wall. Okay, प्रथम देखिए दिखासुन. So it's a complex semi-rigid structure. Semi-rigid means birat besi tight no hoy no. So what is the function? Main function protection. It also prevents the cell from expanding and bursting. Bacterial cell eta expand or burst. It is related with what? We know maximum of the bacteria aquatic region thake. So they have a tendency to absorb water. So basically absorb korile ki hobo phati jabo. So kone prevent koribo? Cell wall. Right? Over. Next point. So when we talk about bacteria, দুটা cell wall or দুখন structure দেখা পাও, এখনক gram positive cell wall বুলি কয়, এখনক gram negative cell wall বুলি কয়. So the cell wall of gram positive bacteria and the cell wall of gram negative bacteria. Gram positive or gram negative or কি কলো difference? It based on the staining process, right? Diagram to আঁকি লোৱা. does not retain crystal violet. মই যে কৈছিলোঁ ষ্টেইনিং ন ষ্টেইনিং বেক্টেৰিয়াক এটা কালাৰ দিব লাগিব ন তেতিয়াহে গম পাম দ্য নেম অফ দ্য কালাৰ ইজ ক্ৰিষ্টেল ভায়োলেট আৰু যেহেতু ভায়োলেট কালাৰ আছে ক্ৰিষ্টেল ভায়োলেট হোৱাৰ লগে লগে সি পাৰ্পল বা ভায়োলেট কালাৰ হৈ যাব ৰাইট নহয় থিন পাৰ্টটো বুলি নহয় গ্ৰাম নেগেটিভ সেল ওয়ালৰ does not retain gram positive cell wall tamane gram positive bacteria bilako crystal violet dile tahat distinguish koribo pariba de microscope ot bhalke sabo pariba color to ami alop pisat porhim ei staining eta bacteria color kyo disu karon ami bacteria bhitoror bostu khini ami নহলে গম নাপাও right jeta tumi color di dia আর যদি colorful হই যায় তাহলে তাক আমি কি বলি কম ইট টেক্স ক্রিস্টাল ভায়োলেট রাইট সো কালারফুল কোন বিলাক হবো ব্যাকটেরিয়ায় গ্রাম পজিটিভ বিলাকে হে এই ক্রিস্টাল ভায়োলেট কালারটো লব কিন্তু গ্রাম নেগেটিভ ব্যাকটেরিয়া এটা যদি মাইক্রোস্কোপৰ তলত চাব পাৰিছা বিচাৰিছা আৰু তাহাঁতক যদি ক্রিস্টাল ভায়োলেট দিয়া তাহাঁতক কিন্তু পানীৰে ধুই দিয়াৰ পিছত তাহাঁতৰ ৰংখিনি গুচি যাব আৰু ৰংখিনি গুচি গ'লেতো আমি এনেও দেখা নাপাওঁ একো সো তাহাঁতক আমি বেলেগ কালাৰ দি কালাৰ কৰিব লাগিব we have something called as saffronin ultimately ইহঁত বিলাক they will appear pink they will appear purple or violet right okay whole so we are talking about the cell wall or ki likhilu innermost layer second point ki likhisu what is the main function protection right third point is bacteria jehetu duta type or no positive or negative he karone tahator cell wall khono positive or negative right so bacteria je is positive bacteria or negative bacteria ase differentiates on the basis of what differentiated on the basis of the cell wall composition right third point jetia ame cell wall boli ko na bacterial cell wall is always made up of peptidoglycan kali class or eta bostu likhisilu whenever we talk about cell wall first cell wall is a characteristic of plant cell and animal cell plant cell second point that we have discussed is plant cell or cell wall is made up of ki cellulose right if you remember we have written a point bacteria cell wall ase the cell wall is not made up of cellulose in its place it is made up of ki peptidoglycan is the point clear peptidoglycan is also called as murin so murin present murin absent murin present in case of bacteria see our peptidoglycan or murin is present clear so if you are talking about positive bacteria তাহাঁতৰ peptidoglycan থাকিব if you are talking about negative bacteria তাহাঁতৰ peptidoglycan থাকিব কিন্তু what is the difference peptidoglycan বেছিকে থাকিব কোট gram positive thin layer কোট থাকিব gram negative is the point clear next what is the difference you see the peptidoglycan আমি সোয়া আমি কেনেকুৱা এটা ডায়াগ্ৰাম আঁকিছিলো এনেকুৱা আঁকিছিলো ন এটো আঁকিছিলো ন অলপ আগৰে স্ট্ৰাকচাৰটো এটো কি বুলি কৈছিল ক্যাপসুল এটো কি কৈছিল cell wall আৰু এটো কি কৈছিল cell membrane আমি কি ডিসকাস কৰি আছো cell wall right so cell wall এখন যদি এটো cell wall খন is made up of কি বুলি কৈছো peptidoglycan মানে এই লাইন স্ট্রাকচার কিনি কৈছে clear so যদি বেছি মোটা হয় positive যদি বেছি মিহি হয় negative clear next peptidoglycan to এটো cell wall inner or পিনে এডাল কি থাকিব cell membrane এখন দেখা তেনেকুৱা হয় নহয় অলপ আগতে আঁকিছিলো ন phospholipid so you have the peptidoglycan layer inner to the peptidoglycan layer what you have the cell membrane is the point clear so gram positive gram negative cell wall or duta difference first peptidoglycan বেছিকে আছে peptidoglycan কমকে আছে second point is in case of gram negative cell wall peptidoglycan etu ki hoy cell wall etu ki hoy cell membrane tahantor cell wall or ekhon extra covering ase juntu niki yat nai eta extra layer pot ase gram negative peptidoglycan yatu ase cell membrane yatu ase right কিন্তু ইহতৰ কি আছে এক্সট্ৰা লেয়াৰ এখন আছে এখানে এক্সট্ৰা লেয়াৰ থাকিলে কি হ'ব এক্সট্ৰা প্ৰটেকশন কি হলে কি হ'ব সোনকালে মাৰিব নোৱাৰা তাহাত ভাইৰুলেন্ট হ'ব এখানে গ্ৰাম নেগেটিভ ব্যাকটেৰিয়া বিলাকে বেছিকে ডিজিজ কজ কৰিব পাৰে right so আমি কি পঢ়িব লাগিব ডিফাৰেন্সেস বিটুইন a gram positive cell wall and a gram negative cell wall we'll be writing the points কিন্তু as of now is the structure understood first bacteria on the basis of cell wall could be of two types right? Bacteria on the basis of different factors amar classification ase. Bacteria on the basis of flagella, keidal flagella ase, corpora ulaise, hekon classification beleg. ঠিক আছে? Bacteria on the type of shapes, hekon classification beleg. Bacteria on the type of cell wall, ekon classification beleg. On the type of cell wall structure, they are of two types. Either it is a gram positive cell wall or it's a gram negative cell wall. What is the difference that you can see from gram positive and gram negative? Gram negative or extra layer eta ase, gram positive or nai. Gram positive or peptidoglycan thicker hoy, gram negative or কম হয়. Another point, gram positive cell wall or a specific bostu present ase. So question ahe, presence of teichoic acid is a characteristic of gram positive ne gram negative? Gram positive. Is the point clear? So heblak ami difference poribo lagibo. Next point that we have studied is this one. Whenever we talk about cell wall generally plantar thake. Plantar cell wall is made up of ki cellulose. Kintu bacteria cell wall is made up of peptidoglycan. Okay? It is teichoic acid or কিছুমান information লিখিছো কিন্তু teichoic acid from MCQ point of view maximum question has been asked about the first point, right? Gram positive or কিছুমান unique characteristics থাকিব, gram negative or কিছুমান unique characteristics থাকিব. For example, অলপ আগতে এটা point লিখিছিলো no, something about pili. Presence of pili is a characteristic of gram negative. Presence of teichoic acid is a characteristic of gram positive. So difference bilak ami পঢ়িব লাগিব, right? As far as the function is concerned, Able functions, it helps in the binding metals kan jote bacteria lagibo pare. Next, it acts as a receptor site for some type of virus. This is a point that you need to know and we'll explain it later also. As far as the third point is concerned, I think it's understood. Degradation mane ki bhangi jua, right? It prevents breaking. So it acts as a receptor site of some viruses. Tamane virus we have an idea, right? Virus e ki koribo pare? Viral attack koribo pare. Virus e kak attack koribo pare? Is it only human beings that suffer from viral diseases? No. It could attack anyone. No? Virus and bacteria attack koribo pare. Suppose we are talking about this as a bacteria, right? And this as a virus. Virus, junto virus and bacteria attack kore, later we will study tahator kisuman naam thake, right? We have a word called as bacteriophage. Pohisa niki structure to ketiaba dekhisa niki enuka eta structure? Dekhisa na? So etu hol eta virus june niki bacteria attack kore. Kisuman virus e আৰু এটা বেলেগ বেক্টেৰিয়া এটাক কৰিব পাৰে কিছুমান ভাইৰাছে বেলেগ এলগি এটাক কৰিব পাৰে ৰাইট সো এইটো কি বুলি কয় বেক্টেৰিওফেজ ফেজ মানে ভাইৰাছ বেক্টেৰিয়া এটেকিং ভাইৰাছ এই ভাইৰাছটোৱে নৰ্মেল বেক্টেৰিয়া এটাক যদি এটেক কৰে এইটো আহি পেলাই ইয়াত মাথা লাগিব লাগিব নেকি ইট হাজ টু ষ্টিক ন সো কোনটো পাৰ্টত ষ্টিক কৰিব কোনে ক'ব টিকয়িক এচিডে ইমানখিনি ডিটেইলছ নালাগে what is the point that is important is this one right I think this is all for today next class on ami ki kore we are discussing certain points about the cell wall টিকয়িক acid লিখিলো second class next class of karone peptidoglycan heading kori thoi dia peptidoglycan boli kole ki jano that the bacterial cell wall or chief component ki peptidoglycan the bacterial cell wall is made up of it is made up of peptidoglycan right what is peptidoglycan we'll talk about it in the next class binding metals means it will bind to the metals also it can bind to anyone no human beings or living or non living or so it helps right thank you however that part is not important this is the only part that we will study right thank you